Thank you for the nice introduction. Yeah, my name is Oliver. I'm part of Timo Dickscheid's working group and Christian's research group now recently. And I'm part of the team working on the 1 micron BigBrain. And maybe some of you remember and were there. 2 years ago, I gave a talk on the next generation BigBrain, which focused on reconstructing the BigBrain at 1 micron, so at cellular resolution. And I brought the first slice from 2 years ago, and I brought this small wish list with me. And the major point was going from optical densities to realistic 3D cell distributions. So actually we're going from the optical density we have BigBrain, we can see where more cells, less cells, to actually seeing how many cells are there, what size are them, the concrete laminar structures. And to achieve this, I will focus on one small part of the reconstruction pipeline, which is artifact correction at 1 micron resolution. What are these artifacts? Artifacts, or especially in this case, processing artifacts, may arise during the pre-processing of the data, so at cutting, staining level. Sadly, these can't be really prevented because the process is too delicate. And as you can imagine, when you see the images on the right, these artifacts can alter the cell count, they introduce maybe false borders in the cell distribution, and so on and so on. So they, in general, hinder analysis. So we want to get some, or generate some data to fill in these missing gaps that we have here. First, the question, how do we actually generate new data? In deep learning, it's quite general statistics. A common approach is to take some noise source and then use some transformation to actually get to your data distribution. This is in general a difficult task to do because it's a large step to go from completely noise, so more or less no information in there, to full information on the image data. And what diffusion models do is break up this hard process into many small steps. So instead of going a large step from full image to only noise, we go to full image with a little bit noise. And this we can remove with, for example, diffusion models, some deep learning network. Then we get some more noise and can remove it. And this way, we go step by step from noise distribution to image distribution. In practice, this works very well. Here I brought two artifacts that you already saw a few slides before. In red, the annotated artifact. And what you will see now when I play the video is the artifact will be replaced by noise. And then over time, the noise will fade out, and instead there will be the new tissue. Now, I don't know how many of you, probably a lot of you have seen the 1 micron BigBrain data. This is purely from the visual perspective very convincing and poses the question, is this actually a plausible cell distribution? Can we actually use this for anything other than visualization? And this leaves us again with the next question. We don't have the cell distribution, the actual cell distribution. How can we check it if that's true? And here I opted to use some proxies for the cell distribution, so some simpler statistics that we can actually compute instead of the actual distribution and compare on those. For the first evaluation, I will focus on purely cell density and cell size. The evaluation workflow is quite simple. We take an image patch that's completely known as a ground truth. We remove a part of the image and then repaint it again. Then, using the CPN, which Eric will talk about later today in a few talks, we can segment the cells in this image patch and then, for example, compute the cell density in this patch or get the distribution of cell size. Comparing these two distributions, we can get a deviation, some score, for example, a relative error, some percentual deviation. And if we do this for enough patches with enough different sizes of occlusion, so how large the artifact is that we simulate, we get this type of graph. On the x-axis we have the masking percentage, so how much of the patch was repainted. And on the y-axis we have the error. And here we can see that the relative error is far below, or is below 8% for the cell size, and even below 5.5% for the cell density. If we go for a simple route, so we, for example, just take the surrounding tissue that we have and infer the cell size and cell density from that for the missing area, the error is between 10 to 20%. So it's quite a big step up. Now this is already quite nice, but not enough, and I already showed you two more proxies that we can look at, or the combination of all, which is basically the cytoarchitecture. So the question is, does the cytoarchitecture stay the same if we repaint? And for this, I used the cytonet that Christian already presented earlier to again repaint a given area, a known patch, put it through the cytonet, and just check for consistent classification in this case. Again, we do this for many patches with different ranges of artifact sizes. And here we can see that for simple consistency, which is here denoted in blue, the K1 accuracy, we have scores above 85%, even if we remove half of the image patch and introduce new tissue there. If we account for small errors, as we've seen for Christian's cytonet, for example, the performance increased a lot by introducing the K2, K3, and K4 accuracy. We achieve almost 100% classification consistency. So these are already very nice results. But when we look at some examples, for example this one, and we inpaint it, there are some problems that appear. For example, wait, I think I can. On the left, on the original patch, we have here a large blood vessel, which is something we would expect to see on the other patch too. As you can see, there is no blood vessel here, or maybe only a small one. In addition, the tissue border also seems wrong, so to say, and the laminar structure is also wrong. So these are all things that the cell statistic is correct, but it's not good enough. And to remedy this, I introduce the neighboring sections, where you can see the blood vessel, of course, extends over multiple sections, so we can actually have the information there that we can use. And if we do this with the neighboring sections as additional information, we can see that it fixes the tissue border, the blood vessel appears, the laminar structure is correct. So overall, this improves the model by another large amount. And I think with these promising results, it's very likely to see that this kind of diffusion model can do the 1 micron artifact correction in the future. And I think I have probably some time left, but I'm already done. Thank you.